Hello students, welcome to Caring Biology Paper 3 Part 2. The first science process skills that we are going to look into today is using space-time relationship. In this using space-time relationship, you are trying to describe the changes in parameters such as location, direction, shape, size, volume, weight or mass in time. For the example, we are going to use this table. Look at the table. We have first column, concentration of hydrochloric acid, and the time taken for pink color of the agar strip to decolorize for the second column, and the third column is the rate of diffusion. And the questions under space-time relationship is, explain the relationship between rate of diffusion and the different concentration of hydrochloric acid. And based on the previous table, we can say that the higher the concentration of hydrochloric acid, the faster the rate of diffusion. Next science process skill is interpreting data. In this interpreting data, you are giving rational explanations about an object, event, or pattern derived from the collected data. As an example, we are going to use this experiment. Look at the diagram. In the diagram, you have test tube X and test tube Y. In test tube X, you have 0.2% starch suspension plus iodine solution plus amylase solution. In test tube Y, you have 1% starch suspension plus iodine solution plus amylase solution. And the questions, based on the results of the experiment, what conclusion can be made about amylase reaction on the different concentration of starch suspension? So student, what is your answer? Okay student, is your answer the same as my answer? For this interpreting data, there are 4 points where you can get the marks. The first point, amylase hydrolyzes the starch suspension. The second point, the final color of the iodine solution becomes brown or yellowish brown. Or you can also answer the dark blue color is decolorized. Third point, when the concentration of starch suspension increases, the rate of reaction increases until it reaches a maximum level and become constant. And the last point, concentration of amylase is the limiting factor. So the suggested answer for the sentences, amylase hydrolyzes the starch suspension, causing the final color of iodine solution to become brown, or the dark blue color is decolorized. When the concentration of starch suspension increases, the rate of reaction increases until it reaches a maximum level and become constant because the concentration of amylase is the limiting factor. Next is defining operationally. In this defining operationally, you are giving meaning to a concept by describing what must be done and what should be observed. Again, we are going to use the same example as the previous example in the video part 1, whereby the experiment is to determine the relationship between the rate of diffusion and the different solution concentration. The agar strip In diagram 1 is prepared by mixing agar powder with sodium hydroxide and phenolphthalein solution that will change color to pink in the presence of alkaline solution. The presence of acid will neutralize the alkali and cause the pink color of phenolphthalein to decolorize. And in this case, the acid we are going to use is hydrochloric solution. And the questions under defining operationally. Based on the results of this experiment, state the operational definition of diffusion. There are two answers which you can use for this question. The first answer is diffusion is the process that causes time taken to decolorize pink agar strip longer when immersed in low concentration of hydrochloric acid. Or you can also use the second answer. Diffusion is the process that causes the time taken to decolorize pink agar strip shorter when immersed in high concentration of hydrochloric acid. Basically, both answer is the same except it is opposite. The first one is a low concentration of hydrochloric acid, and the second one is high concentration of hydrochloric acid. The next one is controlling variables. In controlling variable, you are managing manipulated variable, responding variable, and fixed variable. In a scientific investigation, the manipulated variable is changed to observe its relationship with the responding variable. At the same time, the other variables are kept constant, that is your fixed variable. Again, we are using the same experiment, that is to determine the relationship between rate of diffusion and different solution concentration. Okay, for this experiment, how are we going to control the variables? Okay, for the manipulated variable, the answer would be use different concentration of hydrochloric acid that are 0.2 molar, 0.4 molar, and 0.6 molar. How to handle the responding variable? Observe and record the time taken for the pink agar strip to decolorize using stopwatch. How to handle the constant or fixed variable? Use the same size of pink agar strip, that is 4.5 centimeter. Okay, so the keyword is use different, observe and record, and put your apparatus. And the last one, use the same. Next is hypothesizing, making hypothesis. In this making hypothesis, you are making a general statement about the relationship between the manipulated variable and the responding variable to explain an observation or event. This statement can be tested to determine its validity. Again, we are using the same example. Okay, so we are going to make hypothesis about that experiment. So how are you going to make the hypothesis? The higher the concentration of hydrochloric acid, the shorter the time taken for pink color of the agar strip to decolorize, or the faster the rate of diffusion. And the last science process skill that you need to master is experimenting. In experimenting, you need to plan and conduct an investigation under controlled conditions to test a hypothesis, collecting and interpreting data until a conclusion can be obtained. Below is the flow chart to show the sequence in doing your experiment. We start with identifying the problem. After you have identified the problem, then you formulate the hypothesis. Then you need to identify the controlling variables and also all the other variables. And then planning and carrying out the investigation. Collect the data, analyze and interpreting the data, draw conclusion, and finally writing a lab report. Okay, before I end my video, I will show you what are the necessary things that you need to have in your lab report. Okay, look at this example. Procedure number 1. Measure 2 ml of 0.2% starch suspension with measuring cylinder and put it in the test tube labeled X. Okay, for the step number 1, you will see if you do the measurement, you need to write down the value and you need to write down the apparatus. Okay, and these are what we call as set up the apparatus. Okay, look at number 2. Repeat step 1 for test tube Y with 1% starch suspension. So this one is how you handle your manipulated variable. You repeat but with different concentration of starch suspension. Number 3, put 3 drops of iodine solution into test tube X and test tube Y. This procedure shows how you handle your constant variable or your fixed variable. And number 4, add 1% of 1 milliliter of 1% amylase solution into test tube X and Y. So here you can see 1 milliliter of 1% amylase solution is also your constant variable. Stir the mixture with a glass rod. That one is your precautionary method. And start the stopwatch immediately. And step number 5, record the color change of the solution in test tube X and Y at the beginning of the experiment and after 15 minutes. This one is how you handle your responding variable. So a good lab report should have all these 5 things. The first one is how you set up your apparatus and materials. The next one is how you handle your manipulated variable. The third one is how you handle your constant variable or fixed variable. The fourth one is how you handle your responding variable. And the last one, there should be at least one precautionary method. We have come to the end of the video. So we have covered all the science process skills that you need to know before you take your paper 3 biology SPM paper. So all the best for your paper 3 and good luck.